All right, so we're talking plants and nutrients. So we talked soil and nutrients, but we haven't talked about what the plants do with the nutrients. So why do plants need nutrients? Well, they can't survive on water and carbon dioxide from the air alone. Right? So carbon dioxide used in photosynthesis, but they need other nutrients, right? Other nutrients to build molecules, to make these processes happen, to add structure and support to their tissues. All right, so plants need nutrients, just like we need nutrients, right? Nutrients are going to be substances that might be metabolized or broken down by the plant, or they might be substances that are incorporated into molecules and structures, like I had mentioned there. These different nutrients play many different roles in metabolism. Right? So, plant metabolism, I'm thinking about how they're breaking down and using molecules within their organism. In order to break down molecules, you have to use molecules to help them break it down. Right? So we need a lot of these nutrients. They're structural components of these molecules. The nutrients themselves, we've talked about nutrients coming from the soil. Right? They can also come from water. Think about the soil solution, particularly those anions that are kind of hanging out in that soil solution. But they can also come from the air. Right? So carbon dioxide, plants can take in carbon dioxide from the air. They have the ability to take in some other nutrients, but it's not as big of a resource as nutrients from the soil. When we think about nutritional requirements, right, all these nutrients can be limiting for a plant. Right? If they don't get enough of a nutrient, they're not going to be able to perform X, Y, and Z. Right? All nutrients can be limiting, but we often find that water, right, light, and carbon dioxide are kind of those three main limiters overall. Right? So if they don't get water, they're going to wilt and die. They don't get sunlight, they're not going to be able to go through photosynthesis, they're eventually going to die. Right? They don't get carbon dioxide, they can't perform photosynthesis, they're going to die. Right? But like I said, other elements, mineral elements, can also limit growth, right, or lead to a deficiency symptom. Right? So just like we can be deficient in like vitamins and minerals, plants can be deficient in minerals as well, and they can lead to these symptoms that sometimes we can use to diagnose what's going wrong with the plant. We'll talk about some limitations of that though too. All right, so what are plants made out of? Right? Well, plants are mostly water. Right? They're actually 80 to 90% water. Right? So if you look at a plant that's living, that's actively growing, it's mostly water. Right? If you were to dry this plant, right, stick it in an oven, dry it out, make it nice and crispy, right, what's left over after we've taken all that water out is going to be what we call dry matter. Right? It would be 10 to 20% of dry matter. And out of that dry matter, most of it is going to be carbon, hydrogen, and oxygen. Right? Those are the three most common nutrients we find in plants. Right? The remainder, which is only like 1 to 2% of the plant, will be all those other mineral elements that the plant needs, but doesn't need in such extremes as carbon, hydrogen, and oxygen. Right? Researchers can detect how many nutrients are in a plant using a variety of instruments. Right? One of these instruments that I'm really familiar with is the CN analyzer. It analyzes how much carbon to nitrogen is in a sample. Right? It's a very tedious work. You basically have to ground up all the plants into a really fine, almost powder, and then you make what my instructor told me was these little burritos. So you wrap them up in this tin foil, really, really small. You stick them in there. They'll get essentially like vaporized, and then the contents will be analyzed for the amounts of carbon and nitrogen. It's very tedious, but it's also a very powerful tool because it allows us to see very precisely how much carbon and nitrogen are in our samples. Right? The same type of methods are essentially used in this ICP analysis. But in this analysis, again, we're grinding up the plants, all that dry matter. This time we're going to dissolve it in some sort of an acid, right? Stick it through the machine. It'll vaporize it, right, and analyze the different nutrients as well. Right? So there's some different cool methods that you can use to look at plant tissues. Right? If we look at all the nutrients in general, like I said, the plants need more of some nutrients than another. Right? Those that they require relatively large amounts of, we would call macronutrients. Right? There's quite a few macronutrients. We have them on our chart here. Right? Again, those common ones are carbon, oxygen, hydrogen. But we would also consider nutrients like, did I say carbon, oxygen, nitrogen? Carbon, oxygen, hydrogen. Other ones are nitrogen, calcium, magnesium, phosphorus, sulfur. They're all needed in relatively large amounts. Right? We call them macronutrients. Essentially, this plant cannot complete its life cycle without these nutrients. Right? We can't substitute these nutrients with anything else. Right? So if we're running low on nitrogen, we can't just say, oh, we're going to substitute it with some phosphorus. Right? It doesn't work that way. And it's going to be involved directly in plant metabolism, so the functioning of the plant itself. And on the opposite side of the spectrum, we have those nutrients that are needed in lower quantities. Those would be our micronutrients. They're needed in very small amounts, but yet they're still needed. Right? They're going to function in very specific types of chemical reactions in the plant, make up very specific molecules that are important for the functioning. Those micronutrients might be things like iron and zinc and boron and copper and a whole host of other nutrients here. Right? But we can see that percentage that's actually found in plants' dry mass. So again, we've taken out all the water. They're very, very low. Right? So 0.01% of chlorine is needed. Nickel is needed in I think that's 5 zeros before the 5 there, so 0.00005%. So very, very small quantities. All right, we can classify them based on need. All right, so macronutrients are going to be found in higher concentrations than those micronutrients, like I said before. Right? This is going to vary by tissue. Right? So you're not going to see the same assortment of nutrients in, say, the leaves as you might find in the roots. Right? It's going to vary a little bit. Often when researchers do plant nutrient analyses, we will break it up into leaves, stems, and roots, right? Because we expect to see different amounts of nutrients in each. But what does this mean physiologically? What does this mean for the functioning of the plant? So we can classify nutrients based on their function, not just on how much we need. Right? And we're going to split them into 4 groups. Right? And I've tried to define these groups as broad as possible. Right? We can get into a lot more detail here. But based on their role that they play within the plant. So group 1, we would say that these nutrients are going to be part of carbon compounds. Often they're part of things like amino acids, right, or other compounds that are mostly carbon, right, but they need other nutrients as well. So nitrogen, sulfur are going to be part of carbon compounds. Right, so compounds made mostly up of carbon. The second group here are going to be those nutrients that are important in energy storage. Storing energy to use later, but also in structural integrity. Right? We talked about cells and how they have cell walls, and that provides rigidity and structure to the plant. Right? Especially boron, the molecule boron is really important in cell walls, right? In the structural integrity of our plants. The third group, right, are going to be elements that are simply going to remain in their ionic form. Right? They're not really part of other compounds. They will be used, but they're mostly going to be in their ionic form, like potassium, like calcium, magnesium. Right? Often they're going to be dissolved in plant water. Right? They're traveling throughout the plant. They might be bound to other molecules, but they're often just going to be kind of hanging out. And group 4, right, are those nutrients that are needed in electron transfer reactions. I'm not going to define this too detailed right now. We'll talk about this when we talk about photosynthesis, which is a bunch of these types of reactions. But think about it for now as molecules that are involved in chemical reactions, a specific type of chemical reactions. So things like iron and nickel, right, are needed. Nitrate. Yeah, you could probably pump it in there. I'm really talking about more like the basic elements rather than the compounds like nitrates and nitrites. I always get that mixed up. They're so similar. But these are more like the basic compounds. But yeah, I would maybe pump them in group 4. Yeah. So this is how we might classify them based on how they function. Now when we do the assignment after the lecture today, the plant doctor assignment, I have a couple of slides that you'll just kind of review on your own. That'll give you a little bit more detail on this and will help you classify what nutrient deficiencies might fall into each of these categories. But for now, I'm going to leave it pretty broad. All right. How do we know if a nutrient is essential? Right? Well, we have to determine this. Right? And we're going to determine this through these experiments. And often these experiments are going to be plants that are grown hydroponically. Right? Soils, we've seen it, they're complex. They contain a lot of different particles, and how those nutrients are put into those particles might differ depending on the particle concentration, different porosities, different amounts of organic matter. Really complex. Right? So to simplify things, we're simply going to grow plants in water, right? And water in which we can really tweak the amount of nutrients that we are putting into these systems, right? So often we're going to grow them hydroponically, right? And we might exclude a nutrient and see if the plant grows, how does the plant grow, what's going to occur if it doesn't give this nutrient, right? And we would do this systematically, right? So we include all the nutrients but nitrogen. Or the next time around, we'll do all the nutrients, but calcium, right? And we'll see what happens. So that's how we determine if nutrients are essential. Let's take a step back and do a couple of review questions again. Right? So, what nutrients are most prominent? What might we call these nutrients? And again, just reviewing how we might determine if a nutrient was essential. These are kind of review questions, less applying knowledge questions. I just wrote one. Yeah.
What is the German called?
Yeah. Oh, yeah. 102, right? There's a question about it. That.
I just remember you that like doing it like before the class.
Yeah. I can't remember the name. It's a very common name. Plants adventures and other.
Yeah.
Yeah. All right, I get the sense that everyone's answered this already. We're very quiet. So what nutrients tend to be most prominent in plant cells? What's one nutrient? Carbon, hydrogen, oxygen. Those are the three most common ones. There's a few other of those macronutrients, right? And we said it, they're called macronutrients, right? Each plant need these nutrients, and they are the ones that they need in highest quantities. They're going to be the most prominent. How could we determine if a nutrient was essential? What do we do in an experiment? Yeah, we remove the nutrients, see if it grows, how it grows. Yeah, we study the plant. Yeah, very nice. Any questions? All right, we're going to keep charging along here. I did want to point out that just because there are nutrients available, like say on Mars, Mars has all the nutrients available, right, it doesn't mean that they are accessible for the plant or that the plant can effectively use them. Right? These nutrients might be locked into a form that the plant just simply can't use. Right? Maybe it's the nutrient is there, but there's too little of the nutrients, or too much of a nutrient, which can lead to a toxicity. So, kind of a nice segue into nutrient deficiencies. Right? So, essential nutrients, right? These are substances needed by the plant to complete their reproductive cycle. Right? Plants want to reproduce. Right? Other organisms in nature, we want to reproduce. We want to continue our genetic line, right? We want to make sure that we have the survival of our species. We want to reproduce. And so all those substances by a plant that is needed, we would say those are essential nutrients. The plant needs them in order to pass down their genes to the next generation. Right? When plants lack any of these nutrients, they're going to display some deficiency symptoms. Right? It might be that they might not reproduce. Right? They don't have enough of those nutrients to complete their reproductive cycle. They're lacking an essential nutrient. It might be that the tissues just die. Right? Or the leaves or the other parts of the plants just die off. We might see changes in leaf color. Right? Sometimes we see the leaves might change to like a purple kind of color. Other times they might change to a yellow type of color. Right? This would be called chlorosis. I have a slide on the next slide here, chlorosis, right? This is yellowing of the leaves. Right? And in this particular example, chlorosis is associated with an iron deficiency in this camellia plant here. Right? Chlorosis can be associated with other types of deficiencies as well. Right? Basically, right, the plant or this leaf is slowly dying. Right? It's showing chlorosis. It's showing the yellowing of the leaves. In order to diagnose these types of mineral deficiencies, right, it can be difficult. It can be very difficult, especially when we're talking about plants grown in soil. Because again, soils are complex, right? They might exhibit not just one nutrient deficiency, but multiple, right? So maybe you're trying to diagnose a singular deficiency when in fact it might have two, it might have three. Deficiencies of one, right? So maybe, I would imagine nitrogen is so prominent in plants would be a major deficiency. It might mask the deficiency of another, right? So you might only see the symptoms of one deficiency, when in fact the plant might be suffering from multiple. Right? Also makes it kind of hard to see what's going on. But beyond deficiencies, a lot of viral diseases that plants can get. They can cause the same type of symptoms. Right? So the yellowing of the leaves might not be a nutrient deficiency. It might be some sort of disease that the plant is suffering from. Right? It's hard to determine these nutrient deficiencies. Oftentimes, though, right, it is hard, but we do know what deficiencies should look like if a plant is missing a particular nutrient. Right? So there are often a suite of predictable symptoms, right? Even though it can be hard to sort out those symptoms from other diseases, from other deficiencies. If we had grown a plant and specifically given it not enough nitrogen, right, we would expect a very common symptom to occur, a very predictable symptom to occur. Symptoms are related to the role in the plant, right? So thinking about how those nutrients are used physiologically. part of those group 1, 2, 3, or 4? Are they part of carbon compounds? Are they part of energy transfer reactions? Right? Or do they remain in their ionic form? Right? So it's related to how these nutrients are used. The most common types of nutrients deficiencies you're going to see are nitrogen, phosphorus, and potassium, which is often why fertilizers, right, will advertise specifically the amounts of nitrogen, phosphorus, and potassium that you would see or that you would get from that fertilizer. The most common type. Now, how these symptoms are expressed in a plant are related to something called mobility. So we would say that we could look at these nutrients and classify them as being either mobile or immobile. Right? And so if they're mobile, they can move around in the plant. Right? If they're needed in another area, right, a plant can take it maybe from one leaf and shuffle it to a different leaf. Right? They can be used, they are mobile. Now, immobile nutrients are nutrients that are kind of bound up, they're captured. Right? They can't be moved. If the plant has already used calcium in one part of its tissues, it cannot break that tissue down and move that calcium to another part. Right? That calcium is stuck. You would say it's immobile. And so if you say it's phloem mobile, which phloem is how a lot of these nutrients move, if they are mobile, we'll often see that the bottommost leaves tend to get yellow first, right? Because the plant is taking those nutrients that it needs from the older leaves and shuffling it to the newer leaves. Right? It's mobile, it can be used. If it's immobile, however, right, the new leaves that are grown, and it's kind of difficult to see in this image, but you can see these topmost leaves are really shriveled, they look really unhealthy. Right? The newer leaves grown are going to have those deficiency symptoms. Right? We can't just take the calcium and move it up to those new leaves. Right? All right, so I have a nice illustration to further illustrate the mobility of nutrients, because this is one of the key things in determining, like nutrient deficiency a plant might be suffering from. All right, so if we look at that nitrogen deficiency, right, we have nitrogen in our plant's leaves. If a plant needs that nitrogen elsewhere, right, it can move that nitrogen back into the main part of the plant, maybe it's part of the stem here, and shuffle it to maybe a different leaf. Right? Often it's going to be shuffled to the newly grown leaves, right? The leaves that the plant is actively producing. But calcium deficiency, right? We have calcium in our leaves, they cannot move. Right? They're going to be stuck in place. So if this plant is producing a new leaf, it's often going to look very shriveled, very unhealthy. It's always going to be the new growth that will exhibit these symptoms. All right, so we talked about how do we look at nutrient deficiencies and how it's so hard to do. Right? Talked about whether those nutrient deficiencies, we have to diagnose them from being mobile or immobile nutrients. But if we do want to look at nutrient deficiencies, even though it's harder to do, it can be complex, there are ways to do it. Right? We can take the soil, right, and look at soil nutrient availability. Right? Simply say, the plants are growing in this soil. Does this soil have all the nutrients needed to support life? Right? So we can do some sort of analysis there. The problem again with that is, are these nutrients actually accessible by the plant? We could look at the plant tissues and determine what nutrients are in the plant tissues. Right? So if you would expect high amounts of potassium in the plant tissues, and we run these plant tissue concentrations through our different machinery, right? And we see that there's not a lot of phosphorus, that could be an indication that it's suffering from a phosphorus deficiency. We could do nutrient addition studies. These can be done in the field. We could apply fertilizers to some plants and not to others to see what difference in growth we might expect to see or we might actually observe. But it can also be done in a greenhouse setting. And this is typically more common for nutrient addition studies to be done in a greenhouse study because they're easier to monitor. Right? You don't have the complex interactions of the weather and the climate and other organisms. Right? You just have this very small controlled space in a greenhouse. Right? So you might compare fertilized to unfertilized plants again. Right? This is actually a picture of my thesis study here with my plants. I fertilized some, I didn't fertilize others. Right? You can get really good results from this. Right? They're really nice results. It's very, very labor intensive. Right? You have to be in there every day checking the water levels, making sure everybody has the same amount of soil moisture. You have to be in there every day, just monitoring the plants themselves, taking a variety of measurements. When the experiment is over, you have to grind up all the leaves. You have to run them through the machinery before you can even get the data to give you these types of results. But the data itself is really nice. Right? It tells us a lot about what's going on in the plant. We can treat nutritional deficiencies. Like I said, we can add fertilizers. Right? There's a lot of text on this slide, but I want you to focus on the different types of fertilizers you could add. Right? You could add inorganic fertilizers. Right? So fertilizers that don't have organic material, rather they're going to be like salts. These fertilizers might release a little bit more slowly into the soil. Right? It can limit altering of pH, right? But these types of fertilizers are often leached out of the soil very easily. Right? So the more you water the plants, the more these nutrients will be leached out. We also have organic fertilizers. Right? They're going to come from organic sources, like plant and animal residues. So think about manure being spread along the fields here in Minnesota. They're often a really great way to fertilize a plant, but the plants, or we have to do something to the fertilizer before we can actually spread it out on the field, right? A lot of us here have seen The Martian, right? He uses his feces to fertilize his potatoes, right? In reality, we'd have to decontaminate the feces from anything bad in there before we can actually put it on our plant. We also have something called a foliar application. So instead of sprinkling fertilizers around, right, we can spray the plant down with a fertilizer, and it can be absorbed by the weeds. Right? It's not as common, I would say. Right? I might see this more in maybe orchards. What was that? Yeah. Yeah, harsher environments that the soil's not going to be totally amenable to it. We can actually see that happening right here. Right, we're spraying a plant. These plants are in the Chaparral area of California. So they're pretty hard. Right? They're not great for plant growth. All right, a couple more questions here for us to digest that material. Right, growing a plant in the dorm room. Right? How do you determine what nutrient might be lacking? Right? And what type of experiment could you design if you want to see how a plant responds when it has too much calcium? I know it's talking about it. Yeah. All right, we are growing a plant in our dorm room, and we notice that the new leaves, they don't look healthy, right? Maybe they look deformed or yellow or turning colors. They don't look healthy. We're gonna assume this is from a nutrient deficiency. Right? What type of nutrient do you think this plant might be lacking? You could see that, yeah. And nitrogen, especially, tends to be overall a really common deficiency. I would say in this case, though, it's likely not nitrogen because nitrogen is mobile, and so it could be moved to those newly growing leaves. What other deficiency might be using? It could be calcium, right? It could be other nutrients that are they're not mobile. They're not going to make it to those new leaves, right? And why did you make this determination? Because it's the new leaves that are showing the deficiency, right? Rather than the old leaves. Yeah. Let's see. We want to determine how a plant might respond when it receives too much calcium. What type of experiment could you design? Rather than talking about a nutrient deficiency, we're talking about too much of a nutrient. What can we do? Yeah, we can manipulate the amount of calcium. Yeah. Evan, did we have something to add? Oh, yeah. Yeah, eggshells could be a great addition of calcium. I see a lot of people in gardening provide eggshells. I would also even go on to add that let's assume we have some facilities to work with. Would we want to use soil or hydroponics? Yeah, I like the hydroponics because soil can add some complexity. You can use soil, right? When I did my thesis research, I did a greenhouse experiment with soil because it's easier, less expensive. Yeah. Any questions? All right. All right, so let's talk about plants that might need to conserve nutrients. All right, so maybe they're grown in areas that don't have a lot of nutrients. Right? You might see this when we start to explore other planets, right? That we just don't have the right amounts or the necessary nutrients needed. Plants want to conserve those nutrients, those precious nutrients that they're actually able to get. And so we might see different conservation methods. Right? So maybe these plants are going to have really long-lived leaves. Right? It's going to take a while before this leaf will die and fall from the plant. Right? Here in Minnesota, we tend to see that seasonal. Right? They're going to let go of their leaves every season. Right? But if we want to conserve nutrients, those plants might try to hold on to their leaves for as long as possible. They might also have really slow growth rates. Right? If you're growing, right, you're growing slow and steady, right? You need those nutrients, but you don't need them all at once, right? You're just gonna slowly use those nutrients that are available to you, slow and steady. You're gonna start to grow, right? Rather, plants who have a lot of nutrients, right, they'll often grow really fast, right? They'll use up all the nutrients around them because they don't expect, right? I'm really anthropomorphizing. I cannot say that word. Making plants seem like people here, right? How they're reacting, right? But essentially, they wouldn't expect to see a nutrient deficiency, so they're going to use up all the nutrients around them, grow really fast. We might also see really large nutrient reserves, right? So plants that are grown in these low nutrient environments might have these really deep roots, right? Or a really thick taproot to store a lot of those nutrients, right? The purpose being is that they have a lot of roots, they can explore a lot of area, right? But they can also store a lot of nutrients. So if they don't have nutrients, right, certain part of the year, maybe there's a drought, something is happening, they can rely on what they have stored up. All right, one of the common methods for nutrient conservation is what's called plant nutrient resorption. Right, so resorption is a very common thing to measure. This is the importance of senescing leaves, and senescing leaves basically means leaves that are dying. Right? Leaves that are dying and falling off of the plant. Right? In resorption, right, instead of just letting the plant's leaf fall off, fall to the ground, the plants are first going to take as many nutrients out of those leaves as possible, bring it back into the plant so that they can be recycled for use elsewhere. Right? Now, some plants do this better than others, as we will see here. Normally, the plants are going to drop their leaves in response to like a water stress. Right? They just don't have enough water. They're going to drop their leaves to prevent any more water escaping from them. Right? You might see plants drop their leaves for many other reasons. For example, seasonal reasons. Here in Minnesota, plants are going to drop their leaves because if they don't, the leaves they're going to die anyways because it's going to get too cold. The water inside them won't freeze, rupture the cells, cause a whole host of problems here. And so, resorbing those nutrients or taking those nutrients back in from the leaves can be a very important mechanism for plants that don't generally have access to a lot of resources. Okay? But there are trade-offs. Okay? So it seems like, yeah, why wouldn't every plant just take in those nutrients from the leaves and recycle it? It seems very efficient. Okay? But it can be very costly to do so. Okay? Plants have to expend a lot of energy. They have to use a lot of resources to get those resources back. Right? You have to give some to get some, right? But the benefits, again, is that it's going to limit the amount of nutrients that are going to be lost through those dying leaves because you're bringing them back into the plant. And it's going to decrease reliance on the soil nutrients. Now, you don't have to search out new nutrients. You can use the nutrients that you've already found. Right? But again, it can be very costly to do so. So if you don't need to resorb those nutrients, why bother? Right? If you're going to have plenty of nutrients around, why use that energy? Just get new nutrients. Yeah.
Question on the kidney of leaves.
Mm-hmm.
You know how some plants show you really retained unwanted nutrients and others do not? Is that part of the resorption
I think that would be under resorption if I'm following you correctly. So they're going to shed their leaves to prevent water loss because water can be lost through their leaves.
yes, usually happens during the wet season. They're green so they can absorb as much water as possible.
Mm-hmm.
And then during the dry season, they release so that they don't lose nutrients.
Yeah. Yeah. And if in that process they are taking in nutrients from those leaves they're about to shed, they would be going to resorption. If they're not taking in those nutrients, they just let them fall off, then they want to resorption. Yeah. Does that answer your question? All right. Okay. Yeah, I've never done bonsai pruning before. In fact, I have never really pruned anything before. But yeah, I could definitely see that principle playing off, right? Yeah, or playing out. You cut off leaves, prune them for whatever reason. You're trying to shape the plant. You're trying to prevent something from growing, right? You're gonna have to add those nutrients back in somehow. Right? So you're gonna have to seek them out. Yeah. All right. So we talked about some plants being better at this than others. Right? We can measure this in the form of nutrient resorption proficiency. So how proficient are they in taking those nutrients back in? Right? And to do this, we are looking at the dead leaves, essentially, the leaves that have fallen off the plant. We would collect them, right? Again, we would grind them up, send them through some of our elemental analyzers, and get the results back of how many of these nutrients are left in the dead leaves. Right? If there's a lot of nutrients in those dead leaves, we can assume that they are not good at resorbing. Right? They've left a lot of nutrients in those dead leaves. They have not taken them back in. But if there's not a lot of nutrients, we see really low levels of a nutrient, we can assume that they're really good at resorbing. They have a high nutrient resorption proficiency, right? Because they're not leaving a lot of nutrients in their leaves. Oftentimes, right, these studies have been done before and been done by many scientists, and this work has been published. So if you are doing some sort of nutrient study, right, and looking at those senesced leaves, you can compare your results to published results to see if they differ, to see if they're similar, to make some sort of informed conclusion. I have a nice graph here that I think kind of hones in on this point. Right? So in this graph, we are looking at nutrient resorption proficiency, right, or the percentage of nutrients left in these dead leaves, these senesced leaves, across 3 different species. Right? And we happen to be looking at 2 different nutrients here. So nitrogen and green, phosphorus and purple. Right? First of all, we can see that species B does not have a lot of nutrients left in their dead leaves. This is a good resorber. It's resorbed a lot of those nutrients back into the plant itself and has left very little in their dead leaves. If we look at, say, species C, there's a lot of both nutrients left in those dead leaves. We would say this is a really poor resorber. They're not taking those nutrients in as much. Versus species A, they're really good at resorbing phosphorus in orange, but not so good with nitrogen. And the way that these plants tend to go through these patterns is basically their evolutionary history. They evolved in areas that have low resources. They're going to evolve to be really proficient in taking those nutrients back in. Yeah? I like questions. Ooh. Yeah. I would imagine, so this is definitely going to be species specific as well. Some species are just better at it. So if we're looking at carnivorous versus deciduous trees, yeah, I might expect to see some differences. Right? Coniferous trees, right? So thinking about like pine trees, they tend to grow in more acidic soils, and acidic soils tend to have fewer nutrients. So I could see them being a relatively good resorber, maybe in all nutrients, maybe in some nutrients over the other, depending on that soil type. Yeah. If I were to look at 2 trees in the same species, though, and kind of limit one tree to have these nutrients than the other of the same tree, the same species, I don't know that I would see a big differences in that same species, right? Because it's all about how they evolved, their evolutionary history. They just simply might not have the ability to be a good resorber, even if we gave them fewer nutrients. Yeah. All right. Any questions about resorption? All right, so we talked a lot about nutrient deficiencies, but I did want to mention what happens if you have too much of a nutrient, right? And I've mentioned this in the past, right? Too much of a good thing can be bad. It can lead to toxicity, right? And so if we have toxicity of some nutrients, they're all going to have different effects I'm going to focus on the aluminum toxicity here. Aluminum specifically, if you have too much aluminum in the soil, it's going to inhibit the absorption of nutrients in the plants. Right? It's not going to allow plants to take up nutrients. Right? And aluminum actually does this by negatively affecting the plasma membrane of cells. All right, so we talked about the plasma membrane, the phospholipid bilayer there surrounds all of our cells. Right? Aluminum is going to have a negative interaction with that plasma membrane. It will not allow the plants to take up nutrients as well. And some symptoms we might see with aluminum, but also salt toxicity, or not salt toxicity, iron toxicity here. So aluminum and iron. You might see stunted root growth. So if you look at these two plants here, right, the plant on the right has stunted root growth because it is grown in a soil with excess aluminum. Right? You might see the same if it was grown in a soil with excess iron. Too much salt can be a bad thing too. And too much salt, we often see chlorosis, so yellowing of the leaves. Yeah, that could happen, definitely. I would say in most cases it's not going to inhibit growth to the point where they might die, but they might be very stunted. Yeah, yeah. Maybe you have really bad water, right? And it's going to make the plant die. So we talked about salt, right? Having too much salt can cause chlorosis of the leaves. Plants can get around this, right? Plants can evolve to take up salt, but then exude them or release them back out onto the leaves. So here we see a plant that actually has like this salt crust on it. That's salt that it's taken up from the soil and then exuded it back out into the leaves. They don't want to keep it in the plants themselves because it can be toxic, so they're taking it up and immediately getting rid of it. We can breed plants to be more sensitive or resistant to different toxicities. So these different, like corn to me, different species of corn here, they can be bred to be more resistant to aluminum toxicity. Right? So these have been bred to be more resistant, whereas these are very sensitive to it. So there's different ways we can get around this. Some happen naturally, some happen artificially. Right? We're artificially breeding a lot of plants. All right, time to step back and do another review here. Makes it easier to move here. I'm sorry. All right, let's talk about these questions. Why can leaf senescence be a good thing? Jessica, what is leaf senescence? Oh, Alex, did you hear that? Okay. Yeah, so leaf senescence is basically the death of leaves, right? It happens very natural. It's a natural process. It's a good thing, right? What benefits can leaf senescence have, right? If we drop leaves maybe for a specific reason, why might plants choose to drop their leaves? Yeah, sure. Yeah, they might take the nutrients back in before they drop their leaves. Yeah, yeah. Deciduous trees, that's basically how they prep for winter. We can also resorb, or excuse me, senesce leaves because we might want to prevent any water loss. Maybe it's they're going through a drought, they have to drop leaves so they don't lose as much water. Yeah. What does it mean to be a good resorber? What do plants do when they're resorbing nutrients? Yes, Evan. Yeah, yeah. They're resorber. They're taking up nutrients from leaves that are dying, right? Using it. And typically we're going to find them in areas that don't have a lot of nutrients in the soils, right? They're conserving their nutrients. If we look at plants that are poor resorbers, what type of conditions might we find these plants? Now, I'll look to maybe other areas of the room for this question. Oh, yeah. I like the analogy. Yeah, they have everything they need, right? They don't have to worry about anything. They don't need to conserve nutrients. Why spend the energy to do so? Yeah. All right. I think I'm going to have us take a little bit of a break here. We're getting towards the end of the lecture, and also I forgot my water in my office. I really need some water. So I'm going to have us take, let's do a 15-minute break. When we'll come back, we'll finish the lecture. We'll do the plant doctor activity.
Thank you. Like I would trip if I tried this stuff. I might trip with way during the break. I am trying to find it, but it's like
It doesn't tell you who it is. No.
Well, you'll know if I pause before I say that.
So what I might do is
I can volume. Sounds good. See if I can figure out this quiz situation. Oh. Oh. Funny. No. Yeah, I don't know. Somebody's looking at slides anonymously. Oh, is it? Alright. Is it a lot of snow or just... What? Alright. Okay, I personally, I love cold weather. Hiking in negative degrees. It's, yeah. Exactly, you don't get overheated, there's nobody on the trails, it's peaceful. Winter backpacking? I haven't. It's something I want to try. It's something that's I don't want to do on my own, but it's really hard to convince my husband to do it. So. Yeah. Nice. I've never backpacked in Boundary Waters. I've attempted a through hike on the Superior Hiking Trail. I picked a very bad year to do so. It was like, when did I go? 2000. Was it 2020? No, it was 2021. Yeah. Drought, hot. In fact, we had to pull out because there just wasn't enough water. There's like firemen from New Jersey stationed out there waiting for fires to break out. And in fact, after we left, there was a fire that broke out. So, yeah. Yeah. It was awful. Yeah. Like trying to hike, like through hike up north, you think the weather's going to be a little cooler. Definitely not. Yep. There's no water around, but yet it would still rain just enough to get you wet, but not fill any water sources. Miserable. Yeah. Yeah. Nope. Miserable. Yeah. Oh, very nice. Nice. I've always dreamed of doing at least a section of that. But yeah. Okay. Yeah. Yeah, it does. Yes, I spent a summer in like the Idaho area that was really close to that. Yeah, actually a little south of Coeur d'Alene in Moscow. Yeah, yeah. There, I did an REU program, did some research out there for a summer, but it was such a beautiful area. Yeah. Exactly. Idaho is a beautiful state. Yeah. Colorado. Yeah. Have I? I've been to like eastern side of Washington with and like the Scablands. Yeah. Yeah. Okay. Okay. I can picture that. Woo. Wow. Yeah. I did research in craters in the moon. Very similar type of landscape. Yeah. Mm-hmm. Okay. Yeah, yeah, yeah. Very, very barren, yes. All right, looks like everyone's back. Did anybody check their plants and see new growth? Yes and no. All right. I know. I'm sorry. I don't know why they're growing so slow. I don't know. First time using growth chambers. There's always gonna be maybe. I was actually thinking that'd be faster because in the growth chambers I can set it to 24 hours light, which is what these plants really like. But I don't know. Fun on new things. It's exciting. I just watered them really well this morning because they tend to go through water pretty fast. Yeah, so it's probably why you saw that as something. Yeah, yeah. All right, let's finish up this lecture here. And I want to continue on with talking about plants that are adapted to high nutrients and plants that are adapted to low nutrients. Right? I actually have abbreviations for each of these plants because it is a mouthful to say high nutrient adapted species and low nutrient adapted species. So I often call them HNAPs and LNAPs. Right? You'll see this pretty often. HNAPs, right? They're adapted to high nutrients. They tend to be really weedy species that can grow fast. You don't have to worry about nutrient conservation. They tend to be crop plants. Right? We've engineered crops to grow really fast. That also means they need nutrients, right? But we are providing them with nutrients. LNAPs, right? So low nutrient adapted species. They're used to it, right? They're used to having fewer nutrients in the soil. They've adapted to such, right? You might see things like desert plants or chaparral species, right? They're going to have really slow growth. They're going to have probably a high root mass to search out the soil. They're going to have leaves that don't fall off as often. Like we talked about on one of our previous slides here. So I just want to look at some of the characteristics of each of these. So HNAPs, really fast growth rates. If they get more nutrients, they're going to respond by increasing growth. Right? They're going to store very few nutrients. They don't need to store nutrients because they have plenty of nutrients. They're used to having plenty of nutrients. Their leaves, right? They're not going to be very long lived. Right? They're going to grow their leaves, get the use out of their leaves, drop their leaves. Right? They don't need to hang on to their leaves. They don't need to conserve those nutrients. And they tend to invest less in roots, because why bother spending the energy to explore the soil, right? If you have plenty of access to the nutrients, right? You don't have to explore to get all the nutrients you need. It's all right there. So they intend to invest more into their shoots, into their stems, into their leaves. Right? That's different than our LNAPs. Right? So they are going to grow very slow, very stable, regardless of how many nutrients they get. If they get more nutrients, right, if you add fertilizer, they're not going to respond by an increase in growth. Right? They're going to still grow at the same slow and steady rate. Right? Because again, these plants are grown in an area where they're not expecting to have that continual supply of nutrients. Right? So they don't want to just gobble it all up and use it all in one shot and have nothing left over in reserve. They have a lot of roots. Again, exploring that soil, they're investing in roots. Their tissues, right? So if we look at the concentrations of nutrients in their tissues, they're going to have higher concentrations of nutrients, right? Because they're storing those nutrients. They're keeping them around in case they need them. Right? They're always going to exhibit nutrient storage. Right? So like I said, if we give them more nutrients, they're not going to use it all up right away. They're just going to keep on storing those nutrients. Leaves are going to be very long-lived, like we've talked about before. Right? They're going to stay on the plant for as long as possible to get as much use out of them as possible. And they're going to be really good absorbers. Right? They're going to be really good at nutrient recycling. Alright, so if both of these plants are exposed to low nutrient availability, right, our HNAPs, right, are going to respond more drastically. Right? If they don't have enough nutrients, they're going to reduce their growth, right? Reduce their shoot growth. Right? They have absolutely no nutrients stored up, right? So they can't mobilize the nutrients. They can't move the nutrients from one area to another because they just didn't have the storage of those nutrients. We're going to see decreased tissue concentrations of nutrients because again, they didn't store it. It's not there. They're going to try to uptake more from the soil. Because again, they're adapted to soils with lots of nutrients. They're going to expect there to be lots of nutrients. So they're going to try to get more nutrients. We're often going to see very visual deficiency symptoms. Right? It's going to be relatively obvious that these plants are under duress from lack of nutrients. Right, our LMAPs, if they're going to be exposed to low nutrient availability, right, they're really not going to detect an effect. Right? They're doing their thing, like I said, slow and steady. Right? They have stored up nutrients, so if they are exposed to fewer nutrients than they're used to, they can use those reserves. Right? They can mobilize the nutrients that they have stored up in their leaves to keep up that slow and steady growth. They also have those really large root masses underneath, right? So they can use those roots to help escape any limitations. Right? Use those roots to explore more areas of the soil, try to find those patches of nutrients that might be out there. Right? So long story short, HMAPs, they're going to suffer. LMAPs, they're used to this thing, right? They're just going to mobilize those nutrient reserves that they already have. So, question, right? If we were to engineer a plant to survive in the Martian soil, what types of traits would you expect these plants to have? So think back to what Martian soil has, communications that might be imposed there, and what characteristics of a plant would make it really good to survive in that soil. I'll give you a couple minutes. All right, we have talked a lot about different traits that plants can have depending on if they have lots of nutrients available, if they have few nutrients available. So if we were to engineer a plant to survive on the Martian soil, what might we expect these plants, what traits might we expect these plants to need? likely be a good absorber. Yeah, we know there's low nutrients on Mars. Yeah. What other traits? Would they be an LMAP or an HMAP? Yeah, likely an LMAP, right? Right, Lily, did you have a trait that they would have? Yeah, be affected at nutrient recycling. What might we expect their leaves to look like? Any guesses? Are they going to be flimsy leaves? Are they going to be leaves that fall off easily or stick around? Yep. Yeah. Maybe it's something like a cactus. Maybe they don't have traditionally. Maybe they have spines. Maybe they have leaves that are small and sturdy. Yeah. Yeah. So meats that have traits that allow them to survive these low nutrient conditions, right? So very similar to these LMAPs. Now, what they might look like in reality when we get there, who knows? They probably are not going to be super extreme because likely when we get to Mars, they're going to have to build a greenhouse, right? And they'll be acclimated to some nicer conditions inside that greenhouse, but they probably still have to be accommodated for the lack of nutrients in the soil. All right. All right. My clicker might have died. There we go. So, plant nutrients on the International Space Station. Right? The International Space Station is where plants have been grown in space. Right? And so we're thinking about how the plants obtain their nutrients on the ISS. They're actually using something called a plant grow. Right? They're not using soil. They're not really using hydroponics strictly, but rather they're using these very contained they're called pillows because they have like clay inside of them and like a controlled release fertilizer as the medium for their growth. Right? So it's very interesting how they grow these plants in space. Right? At some point in the class here, I actually have a video of them demonstrating what the plant pillows look like a little bit more in depth here. But it's interesting to see how they do it. There's been some research done on the plant nutrient concentrations that have been grown in space versus the ground. Right? I kind of put the highlights up here. I know it's a lot, right? But just to kind of highlight what they did is they looked at the nutrients available in lettuce crops grown on the International Space Station and compared them to lettuce crops grown in the same conditions but here on Earth. Right? And what they found was that there were differences in the amount of nutrients that were in the plant tissues. Right? So even though they're given the same conditions, there must be something about space that alters the way that plants accumulate the nutrients. Right? But overall, right, they weren't so different to make them unhealthy. Right? They determined that these lettuces, that these leafy vegetable crops, right, were edible and a good fresh food supplement. Right? So there were differences in key nutrients like iron, like potassium, sodium, phosphorus, sulfur, zinc. Right? So there have been some studies done looking at the concentration of nutrients in these plants. Right? So far, nothing major has come out of it. We know there's a difference, but we also know there's safety. Right? We think about plant nutrients on Mars, right? I had to think about that. Likely we're going to need some sort of fertilizer because there's not a lot of nutrients, right? We can only engineer a plant so much, right? But we're going to have to amend the soil in some way. And again, I talked about the Martian and how he uses his feces to fertilize our plants, right? It can be done, right? But they're often going to contain diseases that carry pathogens, which isn't great, right? So you need to be able to properly treat them before you can use them, right? But they do contain many essential nutrients. So yes, this could be an avenue that we could research and look into. Yeah, that's a very good note. Yeah. So his feces have actually been properly treated, right? If you've watched it, they seal it up, they vacuum it, they get rid of all the water, and he has to rehydrate it before he uses it. Yeah. Oh, he deep dried? Yeah, yeah. So they do actually properly control for this in the Martian. The Martian is surprisingly accurate in the science. Um, so we'll talk about it many times, right? We could also think about compost, right? Any food waste that we're using, we could compost it. We could add it to the soil. Though likely there's going to be very minimal food waste because we want to use as much food, eat as much food as we possibly can. We don't want to waste it when resources are scarce. All right, that was the end of this lecture, right? Tomorrow I do have a couple of short videos for you to watch. We will start off class by taking a quiz on it, and then the rest of class is going to be a hands-on activity in the lab, right? There is a note, so I know we moved the exam to Monday, right? But class doesn't start till 11:30 on Monday because of Martin Luther King Jr. Day. And I put a link up there in case you want to check out the events that are happening. And then some upcoming assignments here. Any questions? All right, let's do an activity. So, like I said, we have a plant doctor activity here. So if I change over to Moodle here, there are some slides that I have linked, right? That I'm not going to go through them, but I'll have you peruse them and use them to inform your decisions. And you're going to use that to do an assignment. I linked that assignment on these slides, but that assignment is also up on Moodle here. All right, you're basically looking at images and trying to determine what nutrient deficiency might it have, right? And give me a reason why. And then when you're done, you're going to turn it in here, and that's all I got for you today. So I would encourage you to work together on this. It's nice to bounce ideas off of each other. But this is kind of just a fun do-it-yourself assignment in class. So go ahead.